I'm Dr. Jaimie Hebert, Provost and Vice President for Academic Affairs at Sam Houston State University. At SHSU, we're proud of our legacy. For well over a century, we've been building a reputation as one of the leading universities in a number of different areas, including our world-renowned programs in criminal justice and education. But our namesake, Sam Houston, was a man who never rested on his reputation. The only person ever to be elected governor of two separate states. He also served as both the first and third president of the Republic of Texas. His unwavering passion for leadership and commitment to excellence reminds us that although our past accomplishments may be great, we must always look toward the future. Our legacy at Sam Houston State University is still being built, and the exciting, cutting-edge research being conducted here continues to have major implications, not only for the academic community, but in practical applications that result from it. The projects that are currently being conducted by our faculty and students have profound effects right here in Texas, throughout the nation, even around the world. Over the next few minutes, we'll take a look at just a small handful of the many research programs going on at Sam Houston State University. I think you're going to find the first one quite interesting.
My name is Joan, by the way. I'm the director of the Staffs Facility and associate professor in the Forensic Science Department. I approached the dean of the College of Criminal Justice about a year after I arrived here in 2006. So about 2007, I approached him about the possibility of developing a facility of human decomposition to do taphonomic studies and then ultimately have anthropology, a collection. And the Board of Regents, Tricia Pollard, she was an advocate for it, and so we had some internal funding and we started construction in 2008. The original mission of Staffs was to make it a multidisciplinary facility where students and researchers and law enforcement officers that needed training could come and collaborate on research or training.
My name is Cheryl Rogers and I'm an assistant professor of forensic science here in the department. I have three main projects at the moment. The first is federally funded by the National Institute of Justice, and it's looking at improved tissue preservation for mass disasters. So the NIJ project is looking at preserving human tissues. We are concentrating on skin and muscle samples and preserving them at room temperature. So this is important for our response after a mass disaster, because often the electricity is lost and there is no way to preserve the tissues from all of the victims of the mass disaster. So we may be looking at hundreds or thousands of bodies that are decomposing very quickly. And as that's occurring, the DNA in those tissues is degrading very rapidly, which makes identification of those victims extremely difficult. So what we're looking at is devising a simple method for preserving those skin and muscle tissues without refrigeration, so that we can then quickly identify those remains later on. The second project is looking at improved DNA typing from incinerated teeth. So this is improving the way we can genotype and understanding how we can get better DNA profiles from bodies that have been incinerated, so bushfires, house fires, etc. So Samantha, the student on that project, was at Staffs and removed the mandible and the maxilla from 3 cadavers. And we divided them in half and did half before burning and after burning. We were looking at the teeth, both histologically and what the structure of the teeth are, and then after burning, the DNA identification as well. So what we wanted to identify was, were there particular teeth that were more vulnerable to damage. So therefore we can identify the teeth that would be more reliable for DNA identification after incineration. And also particular regions of the tooth. We wanted to know whether the regions of the tooth, whether it was the crown or the root of the tooth, that was more preserved, so we could get better quality DNA from those particular regions.
My name is Samantha Tippin, and I'm in the Master of Science in Forensic Science program here at SAM. During my first year in this program, I worked as Dr. Hugh Stam's graduate assistant. And so when she came over from Australia, I got to meet her, understand her interests, specifically merging the fields of anthropology and DNA. And so we bounced a lot of ideas between us, and we finally came across the Incinerated Human Teeth Project. So in the past, when there's been a mass disaster such as an arson or a plane crash that involves fire, bones and teeth are the last resort for DNA. So what I'm looking at is focusing on two different aspects of the teeth. One is looking at the cellular structure of the teeth to determine if there's something going on within the tooth that can affect the success of a DNA profile. And second is trying to find a correlation between the cellular structure of the teeth and the success of my DNA profiling.
And the third project is looking at preserving forensically relevant insects that are associated with the decomposition process.
My name is Sybil Buchelai. I'm an assistant professor of biology at Sam Houston State University.
And I'm Aaron Lynn. I'm also an assistant professor in biology at Sam Houston State University. So the project that we're working on really involves looking at what bacteria are present on the human cadaver and how that might change over time through the process of decomposition. But also how is that relevant to the decomposition process in terms of gas production and subsequent insect recruitment.
We place the cadavers out at specific time points, and then through very controlled experimental design, we visit the staff's facility regularly to collect the specimens. We're collecting bacteria and insects and some gas samples as well at these specific time intervals. I like to think it's a very charismatic research project, and we get a lot of we grab their attention with it. A lot of them are interested in biology, but forensics as well. And so we have several graduate students on the project. Some of them are more interested in the microbial side, some of them more interested in the entomology side, and then we have probably a dozen undergraduate research assistants that assist with our graduate researchers. And so the graduate students are, they're generating their master's thesis from this research project. And then the undergraduates, some of them gain research experience and then they go on to different jobs. And then some of them transition into the graduate student role from these projects.
And all the students, I think, are involved in every aspect of the project, especially the graduate students. Our graduate students we have are involved in experimental design, logistical planning, sample collection, data analysis, and they publish and they present at local and regional and national meetings. So student involvement is really high, and they're involved in pretty much all aspects of this research project.
What staffs can offer to the academic community is those students that are interested in chemistry but have sort of a forensic liking or lean towards forensics, or in biology, they have the ability to do research that you're not going to be able to do all over the world. There's only 6 of these facilities in the United States, and most of the world are not permitted to do research on human remains. So in that sense, they have this really unique opportunity to do research. Also, for the criminal justice system, we're able to work with law enforcement to actually using bodies to say, yes, this body's been dead this long based on the research we're doing here. If you use human analogs such as pigs or rabbits, they're not the same. And so this is a very unique opportunity.
While some SHSU researchers are busy studying the effects of death, others are in the business of saving lives. With the help of her students, Sam Houston chemistry professor Alona Petrokovic is investigating better methods to deliver life-saving antidotes to those exposed to cyanide. If she succeeds, her work could save the lives of countless soldiers, military personnel, and civilians.
My name is Ilona Petrikovic. I am an associate professor at Sam Houston State University, Department of Chemistry, and I am the principal investigator of the Cyanide project. Our research is focused on developing a new generation of therapeutic agents to treat cyanide intoxications. The present cyanide therapies are nitidote, that is a combination of sodium thiosulfate and sodium nitrite, and cyanokit, that has hydroxycobalamin as the active ingredient. Why is it important and necessary to develop a new therapy? Cyanides are well recognized as military weapons and terror agents, and the present therapies both have limitations. The limitations for thiosulfate include a relatively low efficacy and unfavorable dispositions, and nitrite can produce methemoglobinemia as a side effect. The cyanokit requires a high injection volume. Basically, both therapies involve intravenous administration that is not suitable for most scenarios. We are searching for new, rapidly acting cyanide countermeasures that can be used in intramuscular injector kits. The research is funded by the NIH and the US Army.
Carbon and nitrogen are two of the most important building blocks of life. Our proteins are made up of amino acids, and they contain both carbon and nitrogen. When carbon and nitrogen are combined, they can, just by themselves, they can form one atom of carbon and one atom of nitrogen and one atom of hydrogen. They can form a compound that mimics oxygen in the body. It's a gaseous compound called cyanide. It's become a modern tool of industry, and so it's used in manufacturing as a precursor in manufacturing many plastics and things. It's used in mining gold. So if you have a wedding ring, probably cyanide helped to bring that out of the ground for you. And it's also been used as an insecticide, and sadly, from that use as an insecticide, it morphed and began to be used as a weapon to kill people. And it's also been used as a terrorist weapon. It's particularly effective indoors. So the current antidotes are designed for administration intravenously, but it's hard to find a vein in everybody, right, if you have a bunch of people down. And so you'd love to be able to have it go into the muscle. Then people could stick themselves, or other people could stick them very quickly, right? But that changes the nature of the problem when you're trying to get an IM, an intramuscular, because now the drug has to go through muscle before it gets into the blood. And so there's a barrier there. And so you have to think of a number of different things. The dosage has to be different. The formulation has to be different. And because there's continuous ongoing research on new ways, better antidotes, this is also an opportunity to use newer molecules that will be more effective than the old ones. Students are extensively involved in this project, both in my lab and Dr. Petrikovic's lab. We have, I would say, probably on average between the two labs of between 10 and 15 undergraduates involved. This is a wonderful example of collaborative research between students and faculty.
My name is Stephen Lee, and I've been working on Dr. Petrikovic's cyanide antidote project. So for the animal study, we first start off by weighing the mice, and then with their recorded weights, we can calculate what volumes of both the antidote that we're going to use as well as the cyanide concentration. And once we've calculated that, we will inject the mouse with the cyanide as a control, and then also cyanide and the antidote as our test. Then we wait and see. So after about 30 minutes, if the mouse has lived or died, we record that. And from that data that we've obtained, we, in our computer program, check them as live or dead and what dose they were injected with cyanide. Then we use what's called the antidotal potency ratio to identify how much protection we get with our cyanide antidote. Currently, Sam Houston has just turned in the patenting papers for our cyanide antidote, so the actual name of it cannot be released. But I can tell you that it's a garlic component and also is an FDA-approved flavorant. It acts as an antidote by donating a sulfur atom to the cyanide to form thiocyanate. So Sam Houston State University has a unique position in that it allows undergrads to do some high-quality research, which is really important in going on to pursue a PhD in chemistry. Whenever I applied for PhD programs, one of the major factors in being accepted to 6 top 20 schools was the research that I performed. Every time then they accepted me, they noted that because of my high level of research productivity as well as my experience that I've gained in both analytical techniques as well as other general lab methods were deciding factors in being accepted into those schools. I'm indebted to Sam Houston the ability to work with such great primary investigators as Dr. Petrokovich and Dr. Thompson.
The hope for this research is that there will be a more effective, rapid-acting therapy for cyanide that can be injected intramuscularly. And this would be especially important in cases where many people have been hurt, where there's been a plant, some sort of a plant spill, like an industrial spill, a big fire in a confined space, a terrorist attack, sort of a military attack. These are the places where we really hope that these new therapies will make a difference.
About 70 miles due south of Dr. Petrokovich's lab is the site of another important research project, the result of which will have major consequences on ecosystems not only here in Texas, but across the country. Let's check in with the Institute for the Study of Invasive Species and find out more.
A couple of years ago, we were looking around to see what we were really good at. And it turned out that we had a lot of expertise in invasive species. And then when we looked around the state and around the Gulf Coast region, we discovered that there wasn't really an invasive species center in this part of the world. It's kind of amazing because, invasive species are such a big problem. Texas is a big state and it's a perfect corridor for invasive species to come into the United States. So just not having a center was kind of remarkable in itself. And then the fact that we had a lot of expertise just made it a natural that we would start to develop a center. And what we've come up with, I think, is really probably the most comprehensive center in the United States. Invasive species are a giant problem in the United States. And in fact, they cause about $130 billion worth of damage a year in the United States. And to put this into perspective, all of the natural disasters together don't amount to $130 billion a year. So invasive species are really just kind of like a natural disaster that is happening throughout the year and not at a single instance like a hurricane or something. So because of that, it's kind of gone under the radar how important invasive species are. One of the invasive species that we're working on is the what was called the raspberry crazy ant. Now it's being called by a lot of people the tawny crazy ant. But this is a species that came into Texas less than 10 years ago from South America. And since its introduction, it has rapidly started to spread throughout Texas. I believe it's now in 26 counties. It's also found in Mississippi, Alabama, Florida. It's just really moving throughout the United States. And in fact, it probably will end up with a distribution something like the red imported fire ant.
I got into crazy ant research when I started my dissertation, and what drew me to it was there was a real need for somebody to pick up the research after my predecessor left. He had done some basic identification and a little bit of management, but besides that, there was nobody else researching this ant. And it was a real problem. The problem with the crazy ant is that they occur in these population densities that are enormous, that can really be described as plague-like proportions. And homeowners are just, they're left feeling hopeless because they don't feel like there's anything that they can do to manage these ants, and really they're right. And so somebody needed to pick up the torch and carry on the research. East Columbia, Texas is where the largest crazy ant infestation that I'm aware of is. That whole community is entirely infested. Not one house does not have the crazy ants.
My name is Cheryl Rogers. My wife and I own the ACOT Cruise Bed and Breakfast. That's what we're in right now. We're in East Columbia, Texas, one of the initial areas originally impacted by the raspberry crazy ants. It can be horrid. We have a pavilion next door that we used to do outdoor events with. At times, the raspberry crazy ants can affect that so much that if food is placed out on a table, within 10 or 15 minutes we're having problems with the food. So it's necessitated an extra level of treatment to try to keep them down. But it's a severe balancing act because on one hand we don't want to poison the earth that we live on, but on the other hand we have to try to find a way to keep these ants so that we can inhabit this area and enjoy this area. We've found times where our water well systems, the points of them have been completely covered with ants. And I don't mean a coating of 5 or 10 or 15 ants. I mean to the point where you lift the cover of the points off of your water well system, and what you have in there is a ball of ants about the size of a tennis ball, just immensely dense. Anything around here that makes electrical contact and emits any type of an ozone attracts those ants. A light switch, a telephone switch, anything. And in that effect, it's made us living here very, very difficult at times.
My name is Cheryl Rogers. I live in East Columbia. I am a permitted state and federally licensed rehabilitator with the Wildlife Center of Texas, which is located in Houston. I primarily do birds and deer. Those are my two specialties. The raspberry crazy ants make a big impact on my caging and housing facilities for these animals. The ants congregate around their water and their food, but they also, even for the high perches for the birds, will go up the perches and harass the animals on those high perches. So with the birds, there's no place for them to go in a caged situation until they're released, and even out in the population, they're going to have to deal with it some, but at least they can move away from it.
Crazy ants, whenever they occur in a population, they're completely ubiquitous. There's not a place that you can step without stepping crazy ants. They swarm over everything, over your feet, crawl up your legs to about your mid-calf, and then run back down. They forage up every single tree, and you can't do anything without them infesting your life. We're at the site of Gaddis and Susan Vityen's house in Lamarck, Texas. They've been dealing with the crazy ants for a couple years now, and they've been very helpful in allowing us to do the research on their property. We've been testing a couple of different management strategies, as well as looking at the flight phenology of the crazy ants. As you can see, they're piling up around the house due to different treatment strategies, as well as the live ants are just crawling over where the dead ants are piling up.
Good morning, my name is Gaddis. My wife's name is Susan, and we've been in Danny's program the last 2 seasons.
Really about 2 years ago, when I was weeding the flower bed, I noticed rows of ants just running crazy. And I noticed that they weren't biting me. They were all over my arms. And so we knew they weren't the fire ants. And then we started noticing we didn't have fire ants anymore. So we contacted our pest control people, and they came out and told us they thought we had crazy ants. And I think they went ahead and treated around the house, but the problem just got worse and worse.
If you'll get a shot of all of the trees that are on this property, and just imagine this at the base of every single one of them, and you'll get an idea of why it's such a challenge to control these ants more than about a month. We can kill a lot of ants, but they're so productive. They produce so many offspring that they just flood right back into the area from untreated areas.
About a week ago, we had been gone on a little trip, and when I came home, the patio was just thick with ants. And when I swept them up, I swept up big piles of them. And when I put them in a bag, a grocery bag. It completely filled that bag up with dead ants. And when you look around, you'll see there are mounds of them, like in the crevices, where sometimes I try not to just sweep them off, because if you allow all the dead bodies to stay, they start to smell bad. So you have to sweep them up and bag them and get them in the trash as quick as possible. You can even see them in the pool. In the summer, it's really sort of yucky because they'll be on the surface of the pool. And then you can get in and sort of, splash a little bit and get that surface tension to make them go down. But you can see it, especially in the hot tub, where they just mound up in there. So it's, it's always another dustpan full of ants every day almost. A couple of the treatments that Danny did, we saw some results for a couple weeks. I think 4 weeks was about the longest that helped, and then it seemed like they just came right back. So yeah.
I asked Danny about the numbers, and one treatment he said, well, I think we've got a 95% good result here. I said, 95%, I did some math, must mean we have a trillion ants. So anyway, even 95%, we were happy with that, but it was still unbearable.
Yeah.
Yeah, outside of the academic community, my goals are to help people. There's a great need for crazy ant management, and there's nothing available to consumers or even to pest control operators that will provide more than about a month worth of control of the crazy ants. So my goal is to help the community.
Now, our idea is that if we can start working on this early and find management strategies for the crazy ant, maybe we can keep it from becoming a problem like the red imported fire ant is. So we've really made this, this research on the crazy ant one of the main focuses of our Invasive Species Center.
Thanks for taking a look at some of our exciting programs. They serve as a testament that the research we're doing here at Sam Houston State University, together with our partners throughout the Texas State University system, is making a real impact on the academic community and around the world.